Okay, hi everyone. We're live again today at 4 p.m. Eastern Time, which was the time voted on by the attendees of the last live workshop. My name is Danielle Snowflack, and I am here to welcome you to another Edvotek live stream. I am the Senior Director of Education at Edvotek, and we're a biotechnology education company. For those of you who aren't familiar with us, today we are going to be focusing on a critical skill in the biotechnology lab, which is pipetting. Now, this is a skill which you're likely using, in doing a lot of different experiments in the biotech lab. But planning for this year is going to be very tricky in terms of which experiments you can run, whether or not you'll be in school or you'll be teaching remotely. How are you going to do your labs? Are they going to be done in person, at home, distance learning? Do you have a hybrid schedule? And so hopefully, some of the stuff we talk about today will help you think about having your students learn about pipetting either at home or in the lab. And ways that you can integrate this. And so we at Edvotek have always prided ourselves on supporting teachers in any way that we can. So today, again, we're going to focus on these options that you can use to help your students learn basic pipetting skills regardless of your model of instruction. All right, so for those of you not familiar with Edvotek, we are the biotechnology education company. We have been around for over 30 years now. We were founded by a professor of biochemistry at Georgetown University, Dr. Jack Trigian. And now in the early mid-'80s, this was a super exciting time for biotechnology. If you think back to the innovations that were happening at this time, we had the advent of the polymerase chain reaction. We had DNA fingerprinting. Scientists were working with recombinant DNA technology to produce pharmaceuticals and bacteria, like insulin. It was the first biologic drug. But little of that excitement in the biology space was being translated into the classroom. And so from there, Edvotek was born. So in addition to creating kits and equipment, we have been active in teacher professional development throughout the entirety of our existence as a company. And this is one way, these workshops are one way that we are working with you, our teachers, to be comfortable with biotechnology education in the classroom. These resources can be used for you to learn about the techniques. These videos can be used for your students to kind of give them some background for the techniques. And so, since our inception, we've been focused on working in the education sphere, where we work with educators all over the world, especially through videos like this, where we can demystify science and help foster the next generation of scientific exploration in our students through hands-on active learning activities. So before we get started, I just want to make one quick announcement. So what you'll see is there is a link in the chat box. So that chat box you should see, on the right side of your screen. So over that way. Feel free to use that chat box to ask us questions, to interact. But what you'll see is I already put a link in the chat box. We are running a survey right now. We want to see how our educators are using our educational resources. It's a quick survey. It shouldn't take more than 5 minutes to complete. If you complete the survey and leave your email address, you will be entered into a drawing for a $50 Edvotek gift card. And so again, the survey focuses on our educational resources. Excuse me. And, we'd love to hear in that survey how they've been useful to you teaching remotely, if you're doing distance learning or hybrid learning, or even how they helped you this spring with everything changing so quickly. In today's workshop, we're going to be talking about the ways that we measure small amounts of liquid in the lab. So we're actually going to be running 2 different pipetting kits today, and with a fun and practical little exercise in the middle that focuses on gel loading. The kits we're going to be running today are the Edvotek MyLab Custom Kit Micropipetting Basics, which is kit 1106. We are going to be also doing a classroom electrophoresis pipetting experiment practice, which is the pipetting by numbers, which is S45, and that's a STEAM activity. So STEAM is science, technology, engineering, A is arts, M is mathematics. So it's taking our STEM and, increasing the real world applications by integrating the arts as well. And so both of these experiments use brightly colored dyes for pipetting. And we're going to combinations of colors. We're going to pipette them onto paper to make some artwork. And it's a really fun hands-on activity to get your students used to using the equipment. And to be clear, we're using non-hazardous dye samples, so there's no biohazard to bring into your home or lab. I was practicing yesterday. I did get a little bit of the dye on my hands. That's the only problem. It did take a little while and a little bit of elbow grease to wash off. But I'll be wearing gloves in the demonstration so that I do not end up with spots on my hands again. So again, they're all food safe dyes, nothing to worry about. Now, the demonstration is going to be recorded. We are recording this demonstration and the slides will be available on our website. And so we are going to put up a survey link for this. So if you would like to get the slides afterwards, what I would ask is that let me copy and paste this. It's a separate Google form that will take you to another form where you can sign up to have me send you the resources, the slides, and the link to the video after the live stream. And so if you fill out that form, we are actually offering professional development certificates to those who watch live. So if you complete that survey, if you would like a professional development certificate, put the checkbox there, and we'll send that to you in the email. I see Brian is saying, "Oh no, what happened? Did something happen? Are we having problems?" Okay, so we are getting a buffering circle. Is my internet okay? Let me try something. Is it getting better now? So there was some problem with rainstorms around here earlier today. I'm wondering if we have some loose wires because YouTube is telling me that you might be getting buffering. Let me check on one more setting to see if I can fix something. This is always the fun of running live workshops. And sometimes there are problems, but I guess that's how we're live. So I hope it is getting better for you. Everything looks okay. I am having, it is mentioning that we have a poor stream on my end. All right, so I think it should be getting better at this point. Okay, good. I think I see Brian. Thank you so much for putting that comment in the chat box. I think I did get an alert on my end too. So there might have been some problems there, but we are back. Please feel free to let me know in the chat box if there are problems. So I was just saying, I put the survey in the link in the chat box. There's a link for a survey where you could enter to win a gift card. And the second survey is going to be about today's workshop. If you would like the slides and a link to the presentation with the awkward interlude and all, we will send that to you. And then if you want, we'll also send a professional development certificate that can be used. That certificate, you will have to complete the link within an hour of this workshop ending. So probably by about 6 o'clock Eastern time, so that we can infer that you were at the workshop. And, and then, I'll email you that certificate either tomorrow or next week. Okay, so enough awkward chatter. Let's get to it. So let's talk about pipetting. So even before we get to pipetting, let's talk about accuracy versus precision. So these are qualities of the measurements themselves. And so if we're thinking about experiments in the laboratory, we want to make sure that they're reproducible, so that every person who does an experiment gets the similar or same results every time they do it. And one way that they do that is, we're making sure we use the same reagents at the same concentrations, but then we want to add the same amount of those reagents to the reaction. And so we need to make sure that we're doing the same experiment every time, basically. And so let's think about setting up a polymerase chain reaction experiment, a PCR experiment. So when you're setting up a PCR sample, basically the volumes of those samples, it's like a large drop of water. It's usually 50 microliters or less, depending on what you're doing. And so within that droplet, we have the enzyme that's going to build DNA. We have our DNA that we're trying to copy. We have primers that target the DNA for amplification. So basically markers for the sequence that we want to amplify. And then we have the building blocks of DNA. So all of these components come together and allow us to make millions of copies of DNA in a short amount of time. But for amplification to be successful, each component needs to be in that tube in the right proportion. And remember, we're dealing with 50 microliters here. It's a large drip of water. And if we add too much or too little of any one of those components, we might not get the results we expect. If we add too much primers, they might bind with each other and give us artifacts. If we don't add enough template, we might not be able to, the assay might not be sensitive enough to pick up the amount of DNA, even though PCR is a very sensitive assay. So pipettes allow us to make measurements and make them reproducible. And so pipetting is such an important technology and technique in the biotechnology lab because it's going to enable us scientists to accurately and precisely measure volumes, which in turn results in successful and repeatable experiments. So in the lab, we're going to talk about let's talk about accuracy and precision in the lab. Accuracy is going to be, excuse me, got a little frog in my throat. In the lab, accuracy is going to refer to how close a measurement is to the true value, while precision is how reproducible that measurement is. So if we're in the lab and we're measuring 20 microliters, for our PCR reaction, and that's the average volume of a raindrop, so that's a really small amount of water, of liquid, accuracy is how close we're going to get to that 20 microliters each time. So, if we're measuring 20 microliters, there might be a little bit of an error. We would be getting maybe one measurement, with one pipette, we get 19.8 microliters. And that 19.8 microliters is pretty close to 20. And it's more accurate than 19 or 21 microliters. So we would say that 19.8, 19.9 microliters, it's a pretty accurate it's a pretty accurate measurement. It's pretty close to where we want to be with our final measurement. Now, precision is how reproducible that measurement is. So again, if we have our pipette and we're trying to measure 20 microliters, and we measure 19.8 microliters, if we were able to pipette that same 19.8 microliters over and over again, that would be a precise instrument. So we would be able to reproduce the same measurement. And so with precision, it doesn't actually matter the volume that we are pipetting. So our measurement could be 20 microliters. If we're only pipetting 5 microliters with that instrument that's tuned to 20, and we get that 5 microliters every time, that measurement is precise, even though it might not be accurate. So that's going to look like that bullseye that's in the top left of our little diagram. Low precision but high accuracy is going to mean we're pretty close to the measurement each time, but we're going to be plus or minus a little bit, but pretty tight to that 20 microliters. Low precision and low accuracy, well, that's just a bad measurement. That's a bad pipette. But we're really aiming for high precision and high accuracy. And that comes down to the tools that we use and the techniques that we use to use them. And we're going to talk about how to accurately use pipettes today. And so in the case of multiple pipette measurements, so if we think about each dot on these bullseye as a different pipette measurement, the set of pipetted volumes will be accurate if their average was close to the intended amount that we intend to measure, and would be precise if the values were all close together. They're accurate and precise if they're both close to the intended volume and close together, and neither accurate nor precise if they don't follow any trend. And so one way to think about accuracy and precision is to imagine a basketball player shooting baskets. If a player has a high accuracy, then the ball will always be close to or in the basket. If the player is precise, then the ball will always go to the same location, which may or may not be close to the basket. So you can imagine if, you could hit the backboard every time, but it just won't go in. Having played basketball in high school, I had plenty of those moments. If you're accurate and precise, you've got Steve Kerr or Steph Curry. You've got like a precision shooter, some of these great great 3-point shooters in the history of basketball. If you're neither accurate nor precise, you've got someone like me sitting home on the couch watching the NBA playoffs, or a team like my New York Knicks who are also sitting at home watching the NBA playoffs. So accuracy and precision of our measurements are going to ensure that our experiments are both successful and reproducible. And so again, I kind of alluded to this, but I have my notes, which are affecting my keystrokes, and it keeps going back and forth. Okay, so I'm also going to go to start showing off our pipetters. So here's my screen. Let me put my gloves on too, just so I don't forget. So pipettes are going to be tools that are going to allow us to measure liquids with accuracy and precision. Pipettes are a large group of laboratory tools that we use to measure and manipulate liquids. The simplest pipettes are going to look like these. So these pipettes are simple plastic tubes with squeezable bulbs at the end. They look like eyedroppers. And they are going to allow us to use suction to move liquids. So if I've got a large jar of water, okay, and I will squeeze this bulb. I will release the bulb, and you can see I have drawn water up into the pipette. To dispense, I am going to squeeze it out. Now, I can use this to measure, this is a milliliter and a half of water. But the measurements the actual markings are just lines. They're not going to be necessarily accurate. They're not going to be necessarily precise for a lot of the experiments that we want to do in the biotechnology lab. So most of the time we are going to use implements like these. So these are going to be our adjustable volume and our single volume micropipettes. And I'll talk more about them in the next slides. But while we have simple pipettes like these transfer pipettes, in a lot of pharmaceutical labs, research labs, we're going to have pipettes that are actually robot driven. So we have these pipetting robots in the lab. And so these robots are programmed to repeatedly release a specific volume to multiple tubes or wells in a process that we would call aliquoting. And I know we've got a lot of teachers in this room. You're probably very familiar with aliquoting, as you prepare the reagents for your students. You may feel like a pipetting robot at times. But these advanced robots can actually carry out entire experiments from measuring and mixing the volumes, examining the results, and even kind of making iterations on the experiments based on the results to try again to get a more successful reaction in the lab. And so we have high-tech pipettes like these robots. We have low-tech like these plastic pipettes. All of them are going to do the same thing. They're going to help us move liquids from one place to the other. But each pipette has a different place and a different time and a different function. So what are we going to be measuring with pipettes? We are going to be measuring liquids like solutions that you may make in the lab. So common chemical and biological reagents that get repeatedly used in most biotechnology experiments are listed in this table, along with several popular reactions and their primary ingredients. So, I mentioned PCR before. That would be our DNA amplification reaction. So the recipe for making DNA in solution would be to take DNA, buffer, our Taq enzyme, and our nucleotides, and mixing them all up together in one tube before performing the experiment. Depending on the procedure being performed, these reactions can range in size from hundreds of liters, which we would think about if we were growing a biological drug in cells in a bioreactor, to 50 microliters or less, like our PCR reactions. So for smaller volume reactions, such as those required for most molecular and microbiology experiments, scientists are going to use micropipettes like these. For biological reasons such as DNA, RNA, and enzymes, the volumes are often measured in microliters. So I have a little table of SI units here. These are, we're in the lab, we're generally using metric units. I'm just realizing now that this chart that I picked was beautiful, but it did not actually go down to the microliter number. I just saw a nice figure that showed both the symbols, the units, and the relation to the liter, but it didn't go as low as microliters, which I wanted to do. Well, take my word for it. There are 1 million microliters in 1 liter of solution. So that's going to be smaller than a milliliter. And this is very small. And so to put it in perspective, again, the 50 microliter sample is, often equal in size to a raindrop. And a raindrop is even large when we're considering the volumes that we're putting together into these samples, which are going to be, 5 to 25 microliters in volume. I know in some experiments I've done, I've even measured as small as 1 microliter of RNA to be used in RT-PCR experiments. And if you tuned in for our last live stream, we did talk about reverse transcription PCR, which is a super cool technique. It's actually used today in research labs to detect COVID-19 disease in people. So precise pipetting when we're doing an assay like that is particularly important because in the microliter range, even small inconsistencies like air bubbles inside of the tip or an extra droplet on the outside of the tip can change the proportions of our PCR, of our reactions, of our experiments. So you can imagine if we're pipetting 5 microliters of a DNA template, and there is a drop on the edge of the pipette tip that's 2.5 microliters, we're adding 50% extra to our reaction, which can really change the results, especially when you're trying to do very sensitive assays. And so in order for your students to have the best possible results in lab, it's critical to make sure that they have the pipetting skills. So most labs today are outfitted with pipettes like these. These are called the piston displacement pipettes. And so I'm not going to take this pipette apart, which I probably could, but I'm not going to because I need it. So inside these pipettes, if we look at the figure on our screen, you can see there is like a cutaway, a schematic of a pipette, of what it looks like on the inside. And so inside the pipettes are a solid disc and a tightly fitted tube. When we press the plunger in, so we're pushing this plunger, this disc is going to move up and down within the barrel of the pipette. And this is going to push air out of the tube and the connected tip. So again, we'll get into this more in depth. But we do use these disposable tips. They prevent liquid from going into the actual barrel of the pipette itself. And they're also disposable, so we're not cross-contaminating between samples. So what we would do is we're going to push the plunger down. We would put this tip into our sample and then release the plunger. And that is the vacuum caused by this piston moving through the barrel of the body of our pipette is going to pull the liquid up into the pipette. Oh, you can't even see my finger. It's going to pull the liquid up into the pipette tip. The volume of this liquid, which is sometimes called our load volume, is equivalent to the volume of air displaced by the downward moving piston. So therefore, load volumes can be set to a specific value by calibrating the piston's height. Early pipettes were like the single volume pipette, where the piston is actually fixed at a single height so that we can only measure one volume with the pipette. However, later displacement pipettes were redesigned so that we can adjust the volume of the micropipette. So this pipette goes from 20 to 200 microliters. And we can actually adjust the height of the piston within the body of this pipette by turning this dial, which allows us to measure a range of volumes. So this one is a 20 to 200 pipette. So, I meant to show this picture. Well, I am showing you the actual pipettes here as well. But the one on top is our adjustable volume micropipette. The ones on the bottom are single volume pipettes. So basically, that pipette on top is able to measure a wide variety of volumes represented by those colorful ones on the bottom. So you could see, the benefits of having an adjustable volume micropipette. You can measure a lot of different volumes with one instrument, with one piece of equipment. All right, so choosing that single volume pipette is easy. If you want to measure 50 microliters, you choose a 50 microliter pipette. This one that we're going to be using today is a 10 microliter. So if you want to measure 10 microliters, you choose a 10 microliter pipette. When using an adjustable volume micropipette, we want to choose one that can measure within the correct range. So what you can see is I have pictures of three pipettes on the screen. I'm holding in my hand in the video a 2 to 20 microliter pipette. So if we needed to measure 15 microliters of liquid, we would want to pick a pipette which flanks the volume, the range volume, the range of that volume. And so 15 is going to be between 2 and 20, so we would use a 2 to 20 micropipette like this. To adjust the volume, so every micropipette is a little bit different in the way they digitally display the numbers that you use to adjust. In this case, our line is our decimal place, so we have 20, so our tens, our ones, our tenths, and our hundredths place. So we can measure, if we wanted to measure 15 microliters, we would adjust the dial until we see 15. So it's going to be 1500. And that's going to be 15 microliters. So we want to make sure to use this micropipette. If we tried to use the 20 to 200 micropipette to measure that same volume, so here you can see the digital display is a little bit different. In this case, we have hundreds, tens, ones, and then tenths place. And again, that line is our decimal place. We could adjust this dial until the digital numbers display 15. But if we are to go below 20, which is its lower limit, we're actually going to break that. We could potentially break that piston mechanism, the mechanism that's going to move that disc up and down to adjust the volume. Breaking the pipette is the worst case scenario. But even if you do dial it below that volume and you are drawing up the liquid, another problem is that measurement is not going to be accurate. So these pipettes are calibrated to work within a specific range. And if we work outside of that range, we're not going to get that accuracy and that precision in our measurements. That is the whole point of using a pipette. So choosing the pipette is just as important to our experimental success as proper pipetting form. And so when you're when you're using the pipette, the first thing to do is actually look at it to see what the range is of the pipettes that you have, and then to take the volume of your sample and figure out which fits into that range. So when you're choosing your pipettes, be sure to choose carefully. So to measure, I'm going to bring out a pipette card. I am in our pipette and some to the end to measure. And you can see this diagram. I'm going to try and demonstrate it as best as possible with my hand here in the screen. Uh-oh, am I buffering again? Can you guys see me? I just saw a little circle. It looked like I was buffering again. Oh no. All right, is my internet? All right, so am I back? It looked like I was getting another error message. I guess with everybody getting back to school in this area, there is a lot of high internet usage. So I hope you're all still hanging with me. I am going to keep trying on. I'm going to keep going on and, pipetting my 17.5 microliters. So what we want to do is we're going to press the plunger down to the first stop. And so if we were all together and we were, and we all had pipettes in hand, I would tell you to push this plunger up and down a few times. And what you can feel, and I hope what you can see, is that my thumb is moving to a position where the plunger is, about 2/3 of the way down. This is our first stop. And if I continue to push, you can see there's a small movement there. That's going to take us to a second point, which is our second stop. And so the one stop is our soft stop, and we can push through that to get to the hard stop. Yeah, thank you. Thank you guys for bearing with me with the internet connection. Again, there have been storms here all day, and I wonder if I'm just having, I wonder if there's just a bad connection. But again, that is I don't want to call it the magic, but that is live streaming. And I'm sure that many of you are teaching remotely or hybrid right now, and so I know you feel my pain. Okay, so I would tell you, so if we were together, I would say, there's these two plunger positions, and these two plunger positions are important for our measurements. Okay, so this first stopping point is the stopping point we're going to use to actually make our measurement, to draw our liquid out. So what I'm going to do is I'm going to push down to our first stop. I'm going to place my pipette tip into the liquid, and so you're going to want to hold your pipette up and down. I'm actually going to do it a little bit sideways so that you can see what I'm doing, but you'd ideally be holding your pipette up and down for your most precise measurements. But what you can see is I now drew out my sample into my pipette tip. Okay, so I did that by pushing to the first stop with my pipette, and then putting my tip underneath the surface of my blue dye, slowly and evenly reducing my plunger, releasing my plunger, and allowing it to come up. And that is going to produce the suction, produce the vacuum that's going to pull the sample up into the tip. Now, to dispense the liquid, we are going to take our tip and we're going to put it on the paper, and we are going to slowly and evenly push our pipette through that first stop to our second stop. And what that second stop does is it pushes a little bit of extra air through the pipette tip to make sure that we get as much of the sample out of the pipette tip as possible. And so what you'll see is that, we have this blue dye. We can see a little bit of it stuck, but that second plunge forces air through the barrel and gets out as much of the liquid as possible. And so that's important. Again, if we are doing something like PCR, where every microliter counts, we want to get every microliter possible out of the pipette tip. And so by pushing that air through, we are pushing out as much as possible to get the best amount of the high maximum amount of sample into our tubes. All right, so that's the basics of using a micropipette. Now let's try our MyLab experiment while we have a good stream health going. Okay, so this is our MyLab experiment. We're going to run the whole thing today, with hoping that the internet is dealing with us. So this 10 microliter pipette actually comes with the experiment. The experiment contains everything you need to do. The kit contains everything you need to do the experiment 3 times. So that's going to be your fixed volume micropipette. We've got disposable micropipette tips in here. Now, unless you have a box of pipette tips at home, you'll want to wash these and reuse them if you're going to run the experiment multiple times. And we wouldn't want to do that if you were doing PCR, because there's the potential for DNA, or, like I'm touching these with my hands, skin cells, oil, to get onto these tips. And that could cause contamination. So either cross-contamination between the samples or contamination from myself, which would give us erroneous results. But for the purpose of pipetting dyes, that is going to be just fine. In this bag, we've got instructions. So the instructions will walk you through the experiment. Let me keep the table up here so you can see it. I think I have it on the next slide too. We're going to have our 3 dyes. So our dyes are red, yellow, and blue, our primary colors. And so we're not using green, so we are colorblind friendly. The reds may look a little different, but we're not worrying that about different hues of green. We have a microtiter plate. These microtiter plate is going to be like mini test tubes where we are going to make our mixtures. And then we are going to have our pipette cards. And so our pipette cards are where we analyze the results. And so this is a paper. You can see it's similar to our grid here. And what I'm going to do, your students will do this. They will cut one of the pipette cards out. And then they will start pipetting and making these mixtures. And so the only thing that your students would need to do this experiment at home would be water. And luckily that is an easy resource to get. So we're going to follow this table to make the dye mixtures, and then we're going to pipette them onto our card to see how accurate our pipetting is. With the included micropipette, which is 10 microliters, we're going to be measuring in multiples of 10. So to make a 50 microliter solution, that is going to be 5 10 microliter measurements. We're going to go through following the table. And when we've finished making all of our measurements, we're going to spot the samples on the card. All right, so I'm going to start with our red dye. And I'm actually using a rack of pipette tips instead of the pipette tips in a bag, just because it's easier than using my hands to try and put them on every time. But again, your students feel for the pipette tips that are included in the kit are just fine for this purpose. I'm only using these because I have them and they're easier to use in this situation. So if anyone has any pipetting questions right now while I'm pipetting, feel free to put them in the chat window. I'm going to start with our red dye, and I am going to pipette 10 microliters into wells 1, 2, 3, 4. We don't put any red dye in there. And then 5, we're going to put 10 microliters. And so one thing you can do with this experiment is you can have your students, make the 5 mixtures and then pipette from these wells, into 3 times into each trial, or you can have them try and make the mixtures 3 times and see if their colors are accurate from measurement to measurement. So for our blue dye, which is going to be our component B, we're going to have 20 microliters of that, and that's going to go into our well 2. And so with the fixed volume pipettes, one thing to mention is that there's only one stopping point, so we don't have that first stop and our second stop. These are going to be a little less accurate than your adjustable volume micropipettes, but again, for most of what we were doing here, in measuring these dyes, they are plenty accurate for what we're doing. So I'm going to measure 20 microliters of blue dye into the second well. If I were reusing this tip, what I would do is I would take it, put it in the water, pipette up and down multiple times. And then what you could see is, that comes, the dye comes out of the tip. And what we'll do is we'll then use the same pipette tip and let us measure our yellow dye. Do I have a paper towel? Let me just get that last little drop of water out of the pipette tip so I don't over dilute our dye. All right, and so we are going to measure 20 microliters of the yellow dye, again pipetting twice with a 10 microliter pipette into this well. And then for our final sample, we'll put 10 microliters here. Does anyone have any pipetting questions? Any thoughts about using pipettes? Are you trying this experiment with your students remotely? I'd love to hear what you're doing. In terms of this training, because this is one kind of training that you can probably do pretty easily remotely. So once you get your students into the classroom, they're already ready to go in terms of micropipetting practice. Again, these are all safe, colorful dyes that can be used at home with no problems. I'm wearing the gloves again because I stained my hands last night when I was going through the preparation for this. But you can, there's no reason why this can't be done safely at home. You can have your students learn how to pipette at home, so that when you have that valuable classroom laboratory time, then those students, you can do the more complicated experiments like the PCR experiments, or gel electrophoresis, different kinds of experiments. And you'll already have gotten this training completed for your students at home. All right, so I'm just measuring the last of the water into our microtiter plate. I'm going to tap it a little bit to mix. You could also pipette up and down to mix it. Either way, we'll mix the samples together. And then what we're going to do is we're going to pipette 10 microliters from each of these wells into each of the 3 lanes. If you want your students to practice making reproducible colors, you can also have them perform these measurements 3 times, and then this would be trial 1. Your second trial would be trial 2. Your third trial would be trial 3. So for me, I am just going to pipette all of these 3 times. Because I'm sure you're eager to get on to the next experiments as well. But I just want to show you, if you're measuring 10 microliters accurately, what you'll see is the dye will spread out to fill nearly the entire circle. All right, so that was our first sample, which was just red dye. Our second sample is a mixture of red and blue dye. So that's going to make us a nice dark bluey purple color. And what you'll see is that over time, this color is actually going to have like a ring form. So it's a very subtle, but what you can see is that there's a light blue ring around like this dark purpley pinkish center. Our third sample is going to be the same mixture as in sample 1. So it's that bright pinky color. And again, that is just to show the reproducibility in the measurements you're making and in creating these different colored solutions. Our next one is yellow, and that's just a straight yellow dye. And this one, and I'll show you this on the next slide. This yellow dye, I feel like, is a great indicator to make sure that your students measure the correct amount of yellow dye into that well. And then our last one is a mixture of red and yellow. Again, I'm pipetting up and down a couple times just to make sure those dyes mix well. And what you can see is we get this nice bright orange color. All right. And that is our completed experiment. And so again, your students can try this a bunch of times. And what you would do is you could have them do this experiment at home, no problem. Take a cell phone picture, scan this, send it to you, and then you can analyze the results this way. And you might think, like, how can I really analyze the results and know that, know how the students actually performed? And so what we would do is you would look at these results. And so again, here are the results that I just got. You can see the different colors. And you can see that I have the picture up there. The colors are a little off in that picture on my screen, but I'm going to show it to you here too. So here's an example of when I did the experiment, but I forgot to add the water. So what you can really see, especially when you're looking at them in person, is that you can see that the red is much darker here in samples 1 and 3 when you don't add the water. So we have a light pink color here, and then we have a darker red color here. And again, I feel like with the yellow, you can see that this yellow is a lot darker. Like all these colors are a lot darker than the colors that you see when you add the water and you have the dilution. Now, we've measured, in this case, we were measuring 10 microliters in the spots. I actually have an adjustable volume pipette here that I used. And what you can see is if we add the incorrect volume, or if we're pipetting incorrectly, we're not either pushing too far when we measure, if we're not pushing far enough when we're pushing the liquid out of our pipette, what you can see is that changes the size of the spot that you're pipetting. And so I can move this out of the way. What you can see is if we only get 5 microliters on, we don't fill up that entire space. If we get 20 microliters on, we are spreading way out from the boundaries of that circle. And remember how I said, with the sample 2, we get that dark purpley center with that light blue ring around it as an indicator that the dye was properly mixed. Well, here you can really very clearly see that like bright aqua colored ring around it. So, any questions about this experiment? Pipetting, micropipetting practice, feel free to ask. I'm going to start moving on to our quick little interlude because one of the things that we do when we pipette is we're actually going to pipette into electrophoresis gels. And so this is one of my favorite things to work on, to use alongside of pipetting practice. These are pipetting, these are DNA DuraGels. And these can actually be sent home with your students as well. They are reusable plastic polymer gels that are the exact same size as our 7x7 electrophoresis gels. But the nice thing is that your students wouldn't have to pour an agarose gel, or you wouldn't have to send an agarose gel home with them. You can send this gel home to them and they can practice gel loading. You can practice doing it right here, just right on the bench top without the bench top, like we're in a lab, right? My basement lab. So right now we are, we are here. And so you can actually practice pipetting right on top of, right on the bench top. You're going to take your pipette tip, put it in the well, and pipette it. But one thing you can do, which I actually prefer, and it gives you a more authentic experience, is to just take that gel and put, here I just have an old lid from a pipette tip container, and we are just going to pour water on top of it. And then your students can actually practice. Oh my gosh, I am pouring water all over the place. I do have a roll of paper towels here just for this problem. All right, let's clean up that water. All right, so now my gel is actually underwater. And what I can do, or your students could do, again at home, is practice for those electrophoresis experiments that they're going to be doing in your classroom. So let me get the bubbles out of the wells. You can see that there are a couple bubbles. You don't want the bubbles in the wells because they're going to affect how your samples go in. But what you're going to do is draw the sample up into the well, and then you will slowly and evenly push the plunger down to dispense the dye into the well. And that lane definitely had a little bubble in it because as I was putting it in, you could see it came out of the well. But there you should be able to see that lane is loaded. And so this is just a nice exercise, a nice practice you can have your students do at home to start learning about pipetting. And then again, when they come to the classroom, when you have time to have them in the classroom, they will have already had some pipetting experience. And so, this might be something you would normally do in class. It's a good example of an experiment that you can do distance or hybrid, depending on what you're doing, to have the students get up to speed, try out loading a gel before they ever get to the classroom, so that the time that you have together really gets to focus on the experiments where you need the equipment that you might not have available at home. All right, so that is our DuraGel. I love these things. And the greatest thing is that when you're done, you can just wash out the wells. I'm actually just going to take one of these plastic transfer pipettes, blow out the dye from the wells. So super easy to clean. Yeah, you can see I had all air bubbles in there, which was affecting how it loads. So you want to make sure to really blow out those air bubbles before loading. You want to clean out the wells after using it. I'm just going to dry it off with a paper towel. If you have a few of these, you can easily sanitize them between students. Send home the gels with a series of students, let them try it at home, bring it back, wipe them down, sanitize them, and then send them to your next student group. No problem, so that they can all try pipetting at home. before your class. All right, so let me move that out of the way without spilling it all over the place. All right, so our third little demonstration, we're going to go through pretty quickly, is we're going to talk about our pipetting by numbers experiment. So this is a fun STEAM application of pipetting practice. So this experiment I love because you're going to be able to do best practices of pipetting. Your students are going to learn the key parts and the importance of accuracy and precision. But then you can actually work together to create sort of a paint by number poster that can be hung in your classroom. And it's a fun activity to have your students practice making measurements, adjusting the volumes of their micropipettes, accuracy, precision, following a protocol, following numbers. And at the end, your students will have worked on something together. So the first thing your students are going to want to do is you're going to want to have them practice pipetting. And so what we're going to do is we're going to take one of our test strips and we're going to take our red dye and we're going to pipette 10 microliters. And again, I'm going to adjust the micropipette to 10 microliters. So right now we're at 19. I'm going to adjust this down until that's 10. It'll look like 1,000, but remember we have 1, 0. The line is our decimal point, and then 0, 0. So I'm going to adjust this down. And again, if anyone has questions, please feel free to put them in the chat window. Here we have 1,000. Again, that means 10 microliters. And what we're going to do is we are going to put a fresh pipette tip on our micropipette. Again, you want to hold the pipette up and down vertical when you're pipetting. So you can see what I'm doing. I'm going to hold it more on an angle. This is not great pipetting form for accuracy and precision, but it will let you see what I am doing a little bit better. So I'm taking my pipette tip. I am pushing down. First thing, I'm going to take my pipette. I'm going to push down to that first stop again. I'm going to put my pipette tip under the surface of the liquid. I'm going to gently release it, which is going to draw the liquid up into my pipette tip. I am then going to slowly and evenly dispense the dye by pushing down to that first stop and then just a little bit and then through to that second stop. And that's going to push all of the dye out of my test strip and onto the circle. And so you want to have your students try and do that a few times just to get comfortable with pipetting and to make sure that their pipetting is accurate and precise. So what I'm going to do is I'm going to try a couple of times. So I'm a molecular biologist by training. It's really hard. Like I feel like pipetting is like second nature to me. It's almost automatic. But what we would do here, I'm going to try and not push the plunger down enough. And so what we'll do is you'll see I didn't push the plunger down all the way. There's still pipette liquid left in the pipette tip. And you can see that the spot is not filling up the entire circle. What I'm going to do this time is I'm going to push my plunger all the way down to the second stop. And I'm going to pull the liquid up. And so we're actually going to be getting extra in here this time. And what you can see is that our drop will expand further past the edges of that circle. If I have the incorrect volume here, so okay, let's go down to 2 microliters. Why not? So I'm adjusting until it's 0.200, which is the bottom end of the range for this micropipette. You can see there's only a very small amount of liquid in there. And when I see that, that's barely a spot. And so before having your students go to the DNA poster, what you would have them do is do these test strips to practice pipetting. And again, here's one that I did earlier. And you can see that we can assess how our students are pipetting just using this strip of paper and this dye based on the size of the circle. When your students get to the point where their dye is filling their circles and they're getting the 10 microliters, then they're ready to go on to the DNA poster. So here is our DNA poster. I started filling this out earlier with our navy blue, which is our 40 microliters. So you can see this comes with a variety of different dyes that we're going to use. Purple. and then we have navy. So there's a lot of navy dye because again, that goes into the backbone of our DNA. But what we would do is if you look at this pipetting card, what you can see is that each circle is a different size, and then each circle in the middle has a number and a letter. So right here we can see this is P30. And so everywhere we see P30, we're going to put 30 microliters of our purple dye. So I'm going to grab my 20 to 200 micropipetter. I'm going to adjust it up to 30 microliters. All right, so we want to make sure it's right at 0300, because again, our top numbers are hundreds, tens, ones, and tenths. And so then we're going to take our purple dye, and then everywhere we see it say P30, we are going to put our dye. And the dye will spread out to fill the circle. And so what you can see is I probably didn't make the most accurate measurement there. You also want to make sure to get the dye in the center of the circle so that it spreads evenly around and it doesn't go out on one edge further than the others. But the kit actually, so this is just one card of the poster. What you can see from the picture is that the poster actually comes with 8 cards. Or no, there's more than that. There's 10. There's 10 cards. So if you have 10 students, you could even potentially do this one at home as well. You'd have to carefully cut around some of the circles or, cut off little pieces of the card where it makes sense. But you could even send small parts of this card home with the students to be able to do pipetting practice. And then when you all come together again, you can assemble the poster. And hang it up in your classroom. And so one of the fun things, that also comes with this experiment is that you can actually do your own pipetting exercise. And so, yes, thank you. I'm glad you think this makes pipetting fun because I totally had fun, especially with this second part, which is to do your own little STEM art. What you can see on the picture is that, the picture that we have up on the screen and then the ones that I did myself is that you can actually take the dye and knowing kind of the rough sizes of the different dye spots and the way the dyes travel, you can make your own artwork. So one of our talented labbies made that awesome shark just by pipetting small volumes in certain areas and large in the other. If you're on the Edvotek email list, you may have seen that Christmas tree, the holiday tree, on the right side of the screen. We actually used that for our Christmas card this year. And I have 2 little pictures that I made last night. This first one started out to be something different, but when I looked at it, it didn't really look like anything. So I just took a Sharpie and made it into a little bouquet of flowers. Here I have my little coronaviruses. And what you could see is I actually put the dots very close to one another so that the edges were overlapping. And you got a little bit of bleed through. So from the blue to the yellow, the purple and the blue, the red and the blue. And it kind of makes like a fun effect. And so you can assess how your students pipette. Again, looking at their accuracy, looking at their precision with the cards, but then they can have fun too and kind of create their own paint by numbers scenario. And so, oh my gosh, it's already 5 o'clock. So I'm going to wrap up really quickly. What I hope this showed you is that pipetting might be something that your students think of as tedious, and teaching it might be tedious, but I really agree. Brian, thank you for saying that in the chat box. This is a really fun way to make, a really cool way to make pipetting fun. We're learning how to make measurements. We are assessing whether or not the measurements are being made correctly. And that's important because making those measurements appropriately is really important when we actually get to the expensive reagents and we're doing those PCR experiments that are time consuming and costly. it's a great way that you could train your students in pipetting at home so that when they get into the classroom that they're ready to go. But it's also, any of these can be done in your classroom as well. So, whatever mode of instruction you're using, these are practices that can be done. So I think, I hope that in the intro I kind of told you about pipetting and why it's so critical in the lab today. I know many of you are doing these experiments already, and so you know how important it is to teach good pipetting skills. And this is a fun way to do it. Plus then you have a nice little picture to hang up in your lab. Different pipettes are used depending on the different circumstances. So we talked again about the adjustable volume, the fixed volume micropipettes. We have some of these plastic transfer pipettes. They're all ways that we can move liquid from one places to the other. And they are tools that allow us to make accurate and precise measurements. And again, learning to properly pipette ensures our accuracy and our precision in our measurements, which is going to contribute to that experimental success. Okay, so we have the link in our chat box. There are 2 links today. Again, one is for a survey on about Edvotek educational resources, how you feel about them. Do you like them? I want to know. Please share in that survey. The second link, again, that I put in the box and Maria also did, that is going to have I'll send you the slides and a link to the Micropipetting Basics workshop video when we're done. Again, if you want that certificate, that professional development certificate, you'll indicate it in that form as well. And there, I will then I'll get your name and I'll send you the certificate. If there are any questions, now is the time to ask. Oh, you know what? Let me get to our last slide. Ignore this link. I never updated the link on that slide to be accurate for the presentation. The link in the chat box is accurate. This is Edvotek. We are in a super cool, I haven't been downtown in a while. We are working from my home studio, otherwise known as my basement lab. But Edvotek is in downtown DC in a super cool building that is a converted news factory. If you need to get in touch with us, we are super accessible. You can email us info@edvotek.com, call us 1-800-EDVOTEK. We are very responsive to our teachers. Another way to get in touch with us is social media. You are here learning about us from YouTube, but we also do Facebook, Twitter, and Instagram. And so if there are not any other questions, I just want to say thank you guys so much for tuning in again. Thank you for bearing with me with the internet. I'm going to try and figure out what was wrong this time. I don't know whether it was the storm that's going through or the fact that school is back in session in Montgomery County and everybody's probably streaming a lot of information. Because we're entirely virtual here. So thank you for bearing with me. I hope you enjoyed this super fun experiment, and I hope to see you or virtually on another live stream or in person eventually next year or whenever we can come together for another science education conference. So thanks a lot. And stay safe. Enjoy, if you're back to school, enjoy your first weeks of teaching. We're thinking about you. And if there's any way we can help, please get in touch with us at Edvotek. Have a fantastic day and we will chat soon. Bye.